Okay, hi everyone. We'll get started here today. It's now 11 o'clock. So my name is Helena Mistry and I'm the Director of Business Development for Imeka. The title of today's webinar is Visualizing Blood Vessel Structure and Function in the Human Brain. Our presenter today will be Dr. Kevin Wittingstall, professor at the University of Sherbrooke. After the presentation, which will be 30 minutes, we'll have a Q&A, and you can either type your question in the chat box that you'll see on the right, or you can unmute your microphone and you can ask. So if everyone could please keep their microphone muted and camera off during the presentation, that would be greatly appreciated. And I'd also like to let you know that we'll be recording this webinar. So Kevin, I hand it over to you and you can start.
Thank you very much, Helena. Do you guys, okay, let me just see. Can you guys see my screen now? Hello, hello,
yes,
can you see my screen?
Yes, we can.
Perfect. Okay, wonderful. Okay, so thank you very much for the invitation. It's a pleasure to be able to present some of the research being done in my lab to you guys today. I see some familiar names in the chat, so nice to see you guys as well. Today's webinar will focus on our attempts at visualizing blood vessels in the human brain, and more particularly quantifying their structure and function. And so just as a primer, many of you probably know this, the human brain is an extremely greedy little organ. Although it's quite small, it consumes a disproportionate amount of the body's total oxygen reserve. So blood is sent to the brain via the large arteries shown here in red. And this blood is then drained back to the heart via the venous system shown here in blue. Interesting fact is that if you take all these little vessel segments and piece them together, it's been suggested that the human brain has roughly a little over 1,000 kilometers worth of blood vessels. And this is because neuronal computation, neuronal calculation requires a lot of oxygen. And since the brain doesn't have the
Kevin, I think you're muted. If you could unmute yourself.
Oh my goodness, I am muted. I've been talking all this time. I'm so sorry. Shall I start from the very beginning?
Sure. We're only 3 minutes in. Go ahead.
I'm sorry about that.
That's okay.
So just as a primer, the human brain is an extremely greedy little organ. Although it's quite small, it consumes a disproportionate amount of the body's total oxygen reserves. So blood is sent to the brain via the cerebral arteries, shown here in red, and is drained back to the heart via the veins, shown here in blue. Interesting fact that if you take all these blood vessels and piece them together, the human brain has roughly 1,000 kilometers worth of blood vessels. And this is because neuronal computation and calculation requires that oxygen be sent to the brain areas that need it in a very, very quick and efficient fashion. And since the brain is incapable of stockpiling oxygen, the way in which this works is that the cerebral vasculature can very quickly actively dilate and constrict in order to efficiently send blood to the areas that need it. And so without going into too much detail, different levels of vessel size are under different types of neuronal control. So for example, the large arteries that ride the surface of the brain here are under control from deep ganglia, whereas the arterioles and the capillaries that plunge into the gray matter are under neuronal and glial control. So you have different types of neuronal messengers here, which is controlling the flow of blood on the surface of the brain and deep within the parenchyma. Now, of course, oxygen is not only needed to power the brain, because of course the brain is never inactive, but if for whatever reason there's a quick stop in blood flow, well, then we get what's called a stroke, where the clinical symptoms are quite acute and quite severe. Now, stroke has been known for a long time, and there are many studies pointing at the role of the vascular system in people who are more likely to suffer from stroke. That's not so much what we do. We're more focused on the fact that in the last 15, maybe 20 years, there's been a lot of data coming out that points to the vascular system playing a role in different neurodegenerative disorders. In fact, one of the main points coming from these studies is that shortfalls in blood flow might actually be one of the earliest biomarkers in these disorders. Now, whether or not this is the cause or effect, we're still not sure. But the idea that the vascular system is somehow impaired in these disorders is now quite clear. And so what's missing, however, is a method of visualizing the health of blood vessels in humans. And so since some of these humans are quite ill, there's a need to do this in a very quick and non-invasive manner. And so an example of what we've done in the past, this was in collaboration with Maxime Descoteaux, where here you see an image of an MR angiography of a healthy subject. It's an axial slice, and in white, what you see here are the main cerebral arteries. I've zoomed in to the left and right posterior cerebral artery. These are the vessels that ensure blood supply to the back of the brain, or the occipital lobe. And here on the left is an image of the two PCA arteries when the subject is resting quietly. And then we take the same image a second time, though this time the subject was watching a movie. So we were visually stimulating the brain. And what you can see is that vessels that were not visible when the subject was resting quietly all of a sudden pop up under visual stimulation. Now, this is because, as I showed earlier, when you send in some visual signals, the neurons in the back of the brain will integrate these signals and send their vasodilatory message to the nearby vessels to cause a local vasodilation, which increases blood volume, flow, and velocity. And that's why we see these vessels here. But what's important is that since we can segment these arteries and now visualize them, we can actually quantify the vasodilatory capacity of these blood vessels. And this is important because in certain neurological disorders, for example, in multiple sclerosis, there was a study that showed that the large arteries in the brains of MS patients have great difficulty in vasodilating, which means that in the event of increased neural activity, the brain might not be able to send the appropriate amount of oxygenated blood to the tissue that needs it. And so by testing the vasodilatory capacity of these blood vessels, it might be a good way to test whether a specific treatment, which is aimed at improving blood flow within a certain arterial zone, is in fact working. And all these images, of course, are without the need of a contrast agent, and they're done using a standard MRI acquisition of roughly 4 minutes. Now, one of the disadvantages of this study is that we only imaged the arteries perfusing the occipital lobe. So we've recently improved on this approach by developing technology to reconstruct the entire vascular architecture of the brain. And we've also added in an approach for segmenting the veins as well. The reason for doing this is twofold. The first is more methodological, in that, without going into details, often when acquiring images that are sensitive to arterial blood, there is always a bit of venous contamination, meaning that it's difficult to reliably say whether or not a blood vessel is an artery or a vein. And so by acquiring the vein image, we can further suppress that contamination. The second reason we do so is that there may be some neurological disorders that primarily affect the venous system. As I said earlier, the venous system drains the blood back to the heart. And so if there's a blockage or a problem in the venous system, well, then this buildup of toxic byproducts could have dangerous repercussions for the nearby parenchyma. And of course, all these images can be easily transformed into different anatomical spaces or different atlases, which means that if we're interested in looking at the vessel density in the hippocampus of a certain patient group, this is of course entirely possible. But the inverse is also possible. By that, I mean the brain is essentially supplied by 3 major arteries. You have the anterior, the middle, and the posterior artery. Each of these arteries are responsible for perfusing different areas of the brain. So, for example, the middle cerebral artery here has a large territory covering the temporal lobe. But each artery is actually physiologically distinct, meaning that it responds differently depending on the type of incoming signals that it's receiving. And so that might indicate that different arteries might be more susceptible to different insults to the brain. And so it's going to be important to isolate the analysis into the different subsets of these arteries when correlating it to clinical symptoms. And so what we've done recently is develop some software to take, again, a standard MRI image of the cerebral vasculature and then automatically segment the vessels, but also give them a label and a name. So this means that we can very quickly isolate a certain vascular segment and analyze the gray and white matter surrounding that particular blood vessel. So for example, I might be interested in automatically extracting the left and right MCA artery, shown here in green and red, and then focus on the main trunk of the MCA. So that's the continuous distribution of the large MCA artery, shown here in blue, and then all of its higher order branches that come off it, shown here in green. And then from this, we can get localized metrics of different morphological features. For example, the local diameter, the length, the curvature, the density, etc. And so to test this, one of the first things we did is we recruited a cohort of young and healthy aging adults. We extracted some of the main cerebral arteries and measured their diameter, which you can see here for the young, and the healthy aging. On average, we found that the cerebral arteries were a bit smaller in the aging population. But the effects seem to be most prominent in the MCA, in particular the second order branch of the MCA called the M2 segment. And you can see that here when you project the results onto an inflated surface of the brain. You can see that the spatial extent of the MCA is larger in the young than the aging. That means that the arteries are reaching the cortex in the young, but less so in the healthy aging. Now, this is quite interesting because we know that, for example, in the case of Alzheimer's, age is of course strongly correlated to the probability of developing this disease. And so if this result is replicated using a larger cohort, it would mean that perhaps the MCA is more susceptible to this than the other cerebral arteries. Now, this is okay for diseases like Alzheimer's, but if we look at different neurological disorders, there's another aspect that pops up that has to do more with the bias of sex. Now, often when we do our studies, we pay a lot of attention of controlling for things like age and different maybe cardiovascular risk. But here I'll show you it's also important to keep track of the male to female ratio in your cohorts. And this is because, for example, as I'll show later, in diseases like multiple sclerosis, this type of disorder affects more women than it does men. And so if there is a vascular component to multiple sclerosis, well, then we need to make sure that there's no bias in otherwise healthy males and females. So what we did is we recruited a cohort of males and females, extracted their cerebral vasculature, and quantified their vascular density, so the number of blood vessels per unit area, in different compartments of the brain showed here in this color map. So the first thing we found is that we reproduced that there were no structural differences in terms of gray matter and white matter between males and females. There was also no differences in the cerebrospinal fluid. When we looked at the venous density map, again, we found no striking difference between the sexes. So here we have the CSF, the cortical gray matter, the deep gray matter, and the white matter. And you see there's no real difference in terms of venous density. But when we looked at the arterial density, we found that women on average had a lower arterial reserve compared to males. Now, this effect is not dramatic, but it was highly significant. And anecdotally, even when you're looking at the images, you can see that there are just fewer blood vessels per unit area in females compared to males. So this is important because when we move on to studying, for example, a disease like multiple sclerosis, where we know there's a strong bias towards females, it's going to be important to keep this into account when we look at our controls. Now, the reason we do vascular imaging in multiple sclerosis is because we feel there are, among others, two lines of evidence that point to the vasculature playing a role in this disorder. The first has to do with the fact that if you look at the distribution of lesions in multiple sclerosis, they tend to accumulate near the horns of the ventricles within the brain, here shown in red. In fact, some scientists have suggested that well over 90% of patients who have lesions will have one or several in these areas. Now, for someone like me who comes more from the vascular side, this is quite interesting because we know that these exact same areas are otherwise referred to as vascular watersheds. So these are areas of the brain that inherently, by design, have a low arterial blood supply. So in other words, it seems that lesions associated with multiple sclerosis would be accumulating in areas of low blood supply. The second reason has to do with many preclinical studies And from what I'm aware of, one post-mortem study in humans where they find large vessel proliferation or angiogenesis in multiple sclerosis patients. So here, for example, is a slide of tissue taken from an otherwise normal appearing white matter in an MS patient, and more or less the same area taken from a normal control. You can see there are more blood vessels in the multiple sclerosis brain compared to the normal control. So we have two lines of evidence here. We have the vascular watershed, which is the accumulation of lesions in areas of low arterial supply. And we have this apparent angiogenesis, or this boost of blood vessel in areas outside of the lesion. Now, since this is usually done using in vivo, a post-mortem analysis, we question whether or not our more macroscopic approach would be able to detect these things in vivo using non-invasive imaging and reconstruction. So we acquired MRI angiographies in 16 multiple sclerosis patients, 16 sex-matched controls, and ran the vascular modeling pipeline through it. Here's an example of an angiographic image from a healthy control, one from a multiple sclerosis patient, and at least by eye, it seemed that we were seeing more blood vessels in the MS patients compared to the healthy controls. And again, when we examine this result over different areas of the brain, of course, always outside of the lesion site, whether we were in the cerebral spinal fluid, so these are in areas that surround the really large blood vessels riding the surface of the brain, or if we were in the subcortical deep gray matter or in the white matter, the result was quite consistent. The vessel density was higher in the multiple sclerosis patients compared to healthy control, thus confirming the previous slide where there seemed to be a larger vessel density in the ex vivo analysis. We then turned our attention to looking within the lesion site. So here in red is an area of the brain where all of our patients had or part of the MS lesion was overlapping in this area. And we extracted the vessel density in the lesion area and compared it to the normal appearing white matter nearby. And again, we confirmed that in the lesion itself, the vascular density is lower than that in nearby normal appearing white matter. Now, it's important to stress that this graph here is not specific to multiple sclerosis. As I said earlier, if you do the same analysis in a healthy control, of course, the healthy control doesn't have a lesion, but if you analyze the vascular density in and around those areas, you're going to get a lower vascular density because, again, by design, these are the vascular watershed. So it seems that these two pieces of data seem to confirm what was once only visible using invasive methods in post-mortem tissue, but now it's encouraging that we might be seeing the same thing in vivo. Now, of course, the question is whether or not this supposed angiogenesis in multiple sclerosis, is it a good thing? Is it a bad thing? We have no idea yet. But I should say that there is a clinical trial ongoing now, funded by the NIH, which is actually looking at administering angiogenesis inhibitors in MS patients. The results are not out yet, but we'll be looking at those with great interest. So, in the end, it's quite clear that the vascular system is playing a key role in neurodegenerative disorders. As it stands, there's not a lot of software that exists for quickly quantifying the structure and function of these blood vessels. We think that this is a critical component in understanding the pathobiology of these different disorders. And so in collaboration with Imeka, our approach can be used to do just this. Our approach is based on, again, relatively short MRI scans ranging anywhere from 2 to 7 minutes depending on the resolution that you want. And most importantly, it does not need the injection of an invasive contrast agent. Preliminary result that I have not shared with you guys today, we're finding that our approach is quite sensitive to MRI scans taken on different MRI scanners, different angles in which the images were taken, and perhaps most importantly, it's quite immune to the actual image quality. So if any of you have old TOF images lying around from a 1.5 Tesla scanner where you think the quality doesn't look that great, please give us a call. We'd be more than happy to collaborate and try to reconstruct these vessels for you. And so, of course, to end, I'd like to acknowledge all the members of the team that make this possible, and of course, all of our funding partners. Thank you very much for your time.
Okay, great. Thank you, Kevin. I would like to open up the floor to questions. So if someone has a question, if you could unmute your microphone and ask, or if you feel more comfortable writing it in the chat box, that's okay too. Does anyone have any questions? Anybody?
Hi, this is Esther from Alcahest. I have a quick question. Thank you so much for this talk. This was actually really helpful. As far as your imaging techniques, I had a question. What size or what's the smallest caliber of blood vessels that you can detect using your technology?
That's an excellent question. I did not mention it. Currently, we are able to resolve vessels that are roughly 0.5 millimeter in diameter. We are exploring some tricks currently in the lab, trying to go a bit deeper. The problem, of course, is that if we want to go lower than about 0.5 millimeter, we're likely going to be limited into the brain region that we want to target. I think it's possible, but we're certainly not there yet, though.
Okay, thank you. And so when you're doing your volumetric averaging, are those then including arterioles? Because I know you said that you take images of the venous system so you can subtract it. And then there's lots of blood still in the smaller caliber blood vessels. How do you allot for that in when you're doing your imaging processing?
Just to make sure I understood the question, how do we ensure that there's no venous contamination in our arterial vessels? Was that the question?
No, it's more of the arterioles. So if you are seeing the caliber of a 0.5 millimeter blood vessel, anything smaller and leading up to the venules, so anything from the venous system to the caliber, how are you then including that in your analysis?
Yeah, so we think that once we're at that level, we no longer see the vessel. So this capillary venous intersection, we're quite sure that we're still far away from that. And so what we really see are the large peel arteries riding the surface of the brain. Once they plunge into the cortex, we might catch maybe the first 0.5 millimeter, and then we'll lose our signal. And then on our venous scan, we'll see the adjoining venule, which is separated by roughly 1 millimeter. So we're pretty sure to capture the neurovascular unit between the two of them at the surface of the brain.
Thank you.
Okay, great. I believe we have a question from Yu-Chen Zhuang on the chat. The question is, what is the MRI sequence to reconstruct the vessel?
Right. So the images I showed you today are based on what's called an MR angiography. Often it's called a time of flight image. This type of sequence is one of the oldest MRI sequences around. It's installed on all MRI scanners. As I said earlier, the way the image is acquired is not really important. Our software can deal with all different types of angles and image resolution.
Great, thank you. We have another question from Sakura Minami. Thank you for the talk, Kevin. Are you aware of this technology being used for any current clinical imaging studies? For example, do you believe this could be used to detect differences between patient treatment groups in neurodegenerative diseases?
Right. So one of the reasons for giving this talk is to try and spread the word. I'm a firm believer in the vascular component of different neurodegenerative diseases. And unfortunately, Sakura, I'm actually not aware of any clinical trial that uses TOF imaging. Typically, TOFs are used to basically scan for patients that might have major infarcts and to basically exclude them from participating in a study. But I am not aware of any trial that actually purposely images these blood vessels in an attempt to detect a narrowing of the vessel, whether it's due to abnormal neural activity or a buildup of atherosclerosis. For me, I think that these things are key elements in the early phase of different disorders. And I hope that with this software package that we've developed, that it will encourage clinicians to include this very, very, very fast MR sequence to their clinical trials.
Thank you. We have another question from Yu Chen. In order to quantify vasodilatory capacity, do you need to induce CO2 challenge or breath holding task?
This is an excellent question. We felt that CO2 administration or CO2 challenge could in fact be challenging for some patient types. And so we wanted to stay away from that based on the potential difficulty of implementing it in a clinical trial. So instead, we decided to go more on the natural route, which is we simply ask our patients to either watch a movie or maybe move their hands as you would in a typical task-based imaging study. And although the differences are not as large as inhaling carbon dioxide, most obviously, we can nevertheless see this vasodilatory response using these everyday tasks. And I would even go further in saying that the movie approach is quite interesting because it does calm the patient or at least reduce some of the stress associated with the MRI experience. So not only are we getting a calmer patient, but we're also getting these vasodilatory responses by simply showing a movie.
Okay, great. Our next question comes from Bambos. Perhaps I missed it, but are there any plans to couple this approach with oxygenation, neurovascularization mapping?
I'm going to try to interpret your question here, properly interpret your question. Of course, measuring oxygenation is the holy grail, and in MRI, it's quite difficult to do so. So on our end, we are not in the oxygenation or at least cerebral oxygenation measurement. In terms of neovascularization, I think we are very interested in this. And so what I showed in the multiple sclerosis is that there seems to be enhanced vessel density in the MS versus the control patients. But your question is quite pertinent because these are two different people, right? So we can't say it's angiogenesis because to do that, we need to take one brain, image it several times, and look in one brain area if vessels are starting to pop out. And this is what we're currently doing in collaboration with the oncology department here at the University of Sherbrooke.
Thank you. Does anyone else have any other questions? I have a question, Kevin. I was wondering if Toft imaging is done preclinically.
It's quite easy to implement. I should say though that in preclinical studies, most researchers will turn to more invasive measures to reconstruct the arteries. But yes, preclinically imaging these arteries is possible.
Thank you. Does anyone else have any other questions?
Hey, Kevin, Pascal here.
Hi, Pascal.
Did you have to build like a standard template for, because how much the brain vessels are different between every individual compared, I mean, we always use MNI template for regular voxel-based morphometry or stuff like that. Did you have to build some sort of template like that?
Yes. So one of my previous PhD students, Michael Bernier, worked very hard in creating a healthy brain arterial and venous atlas. This is free. You can download it along with the source code. If you're interested, please send me an email, and I'll send you the link. So yes, there is a healthy atlas. However, the position of the large cerebral arteries are quite consistent across healthy individuals. They're more or less always in the same spot. But once you get into the second, third, and higher order branches, that's where you start seeing a lot of inter-individual variability. And that's why we think our approach is crucial, because all of our analysis is done in native space. So if we're doing a clinical trial where we want to see how blood vessels respond to a certain pharmacological intervention, you might lose some information when you're trying to fit it into an atlas. So that's why we prefer to work in so-called vessel space, extract the features within the vessel, and then compare that across cohorts, rather than warping everything into MNI and doing the more traditional t-tests.
Okay,
Thank you. Does anyone else have any other questions?
Hi, can you hear me? This is Yutran from University of Rochester.
Hello.
Hi. Yeah, sorry for my question. Ask another question. So I understand you're trying to use the task, ask the subjects to watch a movie or wave hand, kind of use the task of the neural vascular coupling to induce the change. I'm wondering if you have done, or you're aware of any study have done these kind of task-induced neural coupling and the CO2 challenge kind of validation study to compare how much effect these two different challenges are, or how similar they are.
Yeah, that's a very good question. I am not aware of a study that has looked at how much vasodilation is triggered by a sensory stimuli, such as a movie, versus that of a CO2 challenge or breath holding. But I think that, of course, with a CO2 challenge, the dilation is going to be larger. How much larger than that of watching a movie? I can't answer that question. If you're aware of a study doing that, please let me know. But I'm not aware of one, though.
Okay. Thank you.
All right. Does anyone else have any other questions? You're always welcome to write it in the chat box as well. Okay. Well, if there are no other questions, I would like to thank you all for attending. Thank you, Kevin, for your talk. And we'll be sending out a questionnaire afterwards. So if you could fill that out, and I promise it's very short, it's only 5 minutes, and give us some feedback, it'd be greatly appreciated. Thank you for attending our webinar today. Thanks, everyone. Have a great day. Bye now.
Very much.